Hi, this is Kiho Lee from University of Missouri. First, I'd like to thank the organizers for putting together this seminar series. Title of my presentation today is Use of Livestock Models under the Genome Editing Era. Hopefully, with this opportunity, I can introduce some of the activities in my lab. I want to introduce broad level, broad content, so specifics might be missing. Please reach out to me if you have questions. So my lab at University of Missouri really focused on three aspects: biotechnology, embryology, and reprogramming under, I guess, epigenetics. And hopefully I can show you some of the exciting activities in my lab. So we mainly work with pig models. We occasionally use bovine models as well, but pig is our main model. And by using both biotechnology and embryology, we often generate pig models that are relevant in the clinic. So they mimic phenotypes or symptoms of human diseases. And we have a unique model that we utilize nowadays, and those are used for cell transplantation. So livestock models, why do we care about livestock models? Well, livestock models are great for biomedical applications because of similar anatomy and physiology. Pigs are considered to be the ideal species for xenotransplantation, offering organs to human. And pigs show a very similar phenotype as cystic fibrosis. And modeling pregnancy is also pretty accurate with livestock models. Sometimes time matters, and rodents, just their pregnancy just goes through too quickly. But livestock species have a longer gestation period, which makes them an ideal model to study pregnancy and also events of early embryo development. So for agriculture purpose, also these animals are an important source of protein. They provide meat. For example, in 2017, 26.3 billion pounds of beef and 25.6 billion pounds of pork has been produced. So difficulties in using livestock models, why is it? Well, one of the deficiency is low efficiency in genetic modification. So if we have interested in the function of gene A, for example, proper way to elucidate the function is to generate pigs lacking gene A and see what happens to the pig. However, because of the low efficiency in genetic engineering, this has been a challenging task. Instead, people try to get some answers using mouse models because they're widely available. However, because mouse models are not really pig models, even after using mouse models, we have some questions. We can't really get a clear answer to it. Probably in different studies or publications, you hear that mouse models don't really represent human. But also these little mouse models don't really represent pigs either. So what do we do? Luckily, the genome editing technology can solve the issue. It won't solve entirely, but certainly transformed efforts required to generate genetically engineered pig models. And gestation period is another factor that goes into not being able to widely use livestock models. The gestation period is much longer. For example, in pigs compared to rodents, gestation period in pig is 114 days, whereas in mouse is just a few weeks. So there's a big difference. So another thing is just a gestation period, but also breeding time. So gestation period is one thing, but days to reach puberty is very different. In pigs, it's 6 to 7 months. So that means one round of breeding will take over a year. Infrastructure and husbandry, it is really hard to have a facility where you can have lots of pigs. Pigs are pretty big, so that has been a limitation. Most medical schools or even just regular academic settings will have place to house mouse, but if you go ask and, well, we want to raise pigs, that's a different question. So this is a scheme of producing genetically engineered pigs conventionally. So you need to do and previously the genetic information or genome information was not very clear with these animals. So we actually had to go in and clone the genes, doing all these vector constructions and gene targeting, which is very limited using somatic cells. And we do cloning, what's called cloning means nuclear transfer. And we activate these reconstructed embryos, we culture, we transfer them into surrogates to get to make genetically engineered pigs. So cloning efficiency is pretty bad, but historically, efficiency of gene targeting has been near impossible. Thanks to genome editing system, that is not the case anymore. And I don't want to go over too much on genome editing system because the topics are now more or less generalized. But this genome editing system mostly rely on locus-specific DNA break. We do have technologies now that don't rely on it, but most common versions still do rely on locus-specific DNA break. And these genome editing systems has been called as engineered endonucleases or homing endonucleases. As you can see from the name endonucleases, it actually, again, has a nuclease activity. And different versions has been developed, zinc-fingered nucleases, talons, and probably you're most familiar with CRISPR-Cas9 system. It really dramatically changed the efficiency to produce genetically engineered livestock. It really changed all models, but in particular, genetically engineered livestock. We can use this in two routes, and I will elaborate a little bit with another slide in a few minutes. And this is an image of performing cloning, so we can modify somatic cells and perform somatic cell nuclear transfer to generate these animals. Or now, because the efficiency of genetic engineering is so high with genome editing system, we can directly deliver these reagents into developing embryos and still have an impact and alter the genome by introducing these reagents into developing embryos. So again, by using this, we can make special pigs. So as I mentioned, CRISPR-Cas9 system is probably the most familiar ones, and there's a long name. The actual name is clustered regularly interspaced. Short palindromic repeats, it's a long name. And Cas9 is the protein. So it's most recent technology, probably developed around 2012, 13. So it's already been about 10 years. So it's an immune system by prokaryotes. So these bacteria are, although a lot of bacteria cells try to damage us, these phages or viruses can actually damage these prokaryotes bacteria cells. So they actually have an adaptive immune system to make sure once they overcome these infections in a way, they have a mechanism to fight off the phages or virus sequences. The technology really changed the field of genetic engineering because it's much easier to assemble. It only requires engineering complementary reverse RNA as a single guided RNA. And the technology has been actively used in various species, rodents, zebrafish, pig, mosquito, and others. So really prior to CRISPR-Cas9 system, we had a conventional laboratory animals, which are mostly rodents. However, now if you have a biological question that we really want to answer using a unique animal model, there's a way to modify their genome. Variants available, again, other Cas proteins and systems where they can induce genetic engineering, sometimes even without DNA break. So you can see here, so Cas protein recognize the location by guide RNA binding to the target site, and the presence of PAM will let the Cas9 know that this region should be cut and DNA break will happen. So this is a schematic approach to utilize genome editing system. Again, as I mentioned, we can utilize somatic cell nuclear transfer. So once we have culture cells, we can electroporate typically, but we can use other means to transfect CRISPR-Cas system into these cells, and we can select them. And then by performing nuclear transfer, we remove nuclear material of an oocyte, and then introduce these donor DNA or genome of donor DNA, and that's how we generate these animals. So we generate cloned embryos, we culture them, and then we transfer them into a surrogate, and that's how we get these piglets, as you can see here. Another approach that has been developed because of genome editing system is direct injection. What it means is we can generate embryos through in vitro fertilization or other means, and then directly inject the embryos with CRISPR-Cas9. So as you can see in this figure here, so we can directly poke embryos and culture these embryos, and same thing, we transfer these embryos into surrogate, and that's how we get genetically engineered piglets. So both systems have pros and cons, advantages and disadvantages. We won't probably go over those in details, but it's a very dynamic field to design specific strategy and generate interesting models. So first model that I would like to introduce is production of immunodeficient pigs for cell transplantation. So use of SCID pigs, so these are severe combined immunodeficiency pigs for regenerative medicine. So it's almost getting 10 years ago, but 2014, I demonstrated that these pigs can support growth of human stem cells. And this is an image of pig lacking adaptive immune system. And when we introduce human stem cells into the pig, it was behind its ear, and we were able to see the cell growth or tissue growth out of these stem cells and what's called teratoma. So teratoma was formed, and you can see the image of teratoma in this petri dish. And SCID pigs can also be used to humanize pig models. So we can, by introducing human lymphocytes, we can make human antibodies. It could be cancer model. We can grow cancer in these pigs. HIV study, because they don't have immune system, the model can be effectively used to study HIV. Stem cell therapy and organogenesis. We are mainly using these pigs for either cancer application or try to generate human tissues that can transplanted into a patient. So first report from 2014. So we modified a gene called RAG2. So at that time we used genome editing system, what's called TALEN, and we used those sets to modify the genome in somatic cells. And at that time, this was a very huge increase in genetic modification. Now we can probably do better, but knockout efficiency was 40%, and 14% of cases we were able to modify both alleles, so two alleles in somatic cells. So these are the known genotypes that we identified, and we use these donor cells for nuclear transfer to obtain pigs that lack functional RAG2. So again, these were through cloning, and one of the features of immunodeficient animals is the lack of thymus. So when we did necropsy on these animals, and you can see here, these pigs did not have thymus, and whereas heterozygous pigs and wild-type pigs have the thymus formed, and spleen where a lot of lymphocytes are developed, as you can see here. Its spleen is very underdeveloped compared to their controls. And again, this image shows, although it shows some thymic structure, this was the only pig that we can capture the presence of thymus. But you can see here from compared to the wild type, the size of thymic looking structure was very small. And we also performed immunohistochemistry histology and immunohistochemistry to identify lymphocytes, so B cell markers, T cell markers, and NK cells. Because the lack of RAG2 impacts adaptive immune system, you can see here no B cells, no T cells. NK cells, which is a part of innate immune system, was still there in the pig. And we looked at the level of B cells and T cells in their spleen. So splenocytes were isolated and ran through FACS. And you can see these pigs lack immune system, so adaptive immune system. So next, what we wanted to do is transplant human stem cells and see what happens. And you can see here that's a teratoma formed. And when we ran histology, we definitely saw cells representing different origin. And what it means is we saw cells representing different germ layer, ectoderm, endoderm, and mesoderm, which reflects the type of cells that derives the entire body. So the fact that the teratoma contains all three germ layers indicates that this pig could support growth of all the cell types of human. We ran another immunohistochemistry to follow all these markers, and we saw many markers in the teratoma, again indicating that these stem cells, and these were human iPS cells, induced pluripotent stem cells. So these pluripotent stem cells can differentiate into different cell types. So this was the first report indicating that these SCID pigs can support growth of human cells. So the next task we had was to generate SCID pigs lacking both adaptive immune system and innate immune system. And this was the first report. And so our thought was to disrupt both RAG2 and IL-2 in this pig, because that will make these pigs lack all major lymphocytes, so that their phenotype will be similar to NSG mouse. And these pig models will be ideal for stem cell transplantations, because they would allow growth of human cells and tissues. But these pigs will be very tough to make them through breeding, because, again, immunodeficient pigs, they don't have immune system, so they don't really survive. They don't really live long enough to be used for breeding. So I guess one possibility is to maintain them at heterozygous fashion or hemizygous fashion and use those for breeding. But we tried to design that scheme. But then that really told us that we need to make 256 pigs before we generate these NSG-like pigs. So after looking at that number, we said, oh, there's no way that we can produce these through breeding. So we decided that, okay, what if we just generate these immunodeficient pigs without breeding? And that's where we decide to use technology, famous technology, CRISPR-Cas9 system, to disrupt both RAG2 and IL-2 simultaneously in developing embryos. So this was the first report of NSG-like SCID pigs. This was published in 2016. So we designed a CRISPR system that can disrupt both RAG2 and IL-2. And we generated these CRISPR-Cas9 tools in the lab, and we did microinjection. So this is an embryo. We poked the embryos, and we transferred them into surrogate pig. And this is an image of first SCID pigs that we generated through this approach. This was the birth of first, I guess, full SCID pigs, or NSG-like SCID pigs, if you will. So similar to RAG2 knockout pigs, they did not have thymus. So that was a clear phenotype we saw right away. And then when we looked at their lymph nodes, so compared to wild type, as you can see here, this is an image of the entire gut area. You can see these pigs do not have lymph nodes. So again, indicating their immune system is compromised and they don't have much lymphocytes. So we ran FACS to verify this, and we took cells from the gut and circulating blood, and you can see here, again, the level of B cells, T cells, and NK cells. They were all suppressed in these SCID pigs compared to to wild type. So that really demonstrates that we have successfully generated these pigs. We also did some immunohistochemistry to really see the presence of B cells and T cells and NK cells, and you can see in this histochemistry, so you can see the red signals indicates B cells, T cells, and NK cells. You don't see those cells in these pigs. So that really tells us that they do not have functional immune system. And you can see here in the spleen, what's called white pulp, a germinal center where they generate lymphocytes is really poorly developed in these pigs. Again, so white pulp, so no germinal center, no white pulp, no B cells, no T cells, no NK cells. So now that we have these pigs, our next question was, can we use these pigs for human stem cell transplantation? So again, the format of this plan is that if we could get patient-derived xenografts, so if we can take cells from patient and to introduce into these pigs, and induce these cells to differentiate into mini organ, if you will. So we have liver or intestine developed from these pigs. Then because these organs or tissues, if you will, are developed from a patient, that mini organ can go back to the patient without immunological rejection. So that's the idea of performing in a way, ex vivo generation of organs and going back to a patient. Another application of SCID pig is to induce or transplant human cancer cells so that these pigs will present a human cancer and then use those to monitor and monitor progression of cancer or develop new treatments to battle cancer. Because these pigs lack immune system, we need to have very high biosecurity to make sure we can keep them alive. So one study I would like to introduce is transplantation of human organoids. So these organoids are 3D structure, and it represents characteristics of intestine. And we did a transplantation surgery, and at the end of 3-4 week mark, we were able to harvest human cells or human tissues that are not rejected in these pigs. So these tissues resemble human intestine, and again, this demonstrates that these pigs can be used to grow human organ, and in theory, ideally, that organoid tissue can go back to a patient. The second application is cancer treatment. So by working with oncologists, what we did is we introduced human pancreatic cancer cells into our pig model. And this is tumor growth that we captured as time goes. And then these pigs with right care, they were thriving as well as we measured their weight, and they were growing. And you can see here, so you can see compared to control pig, you can see the formation of tumor here. And then actually this was detectable using ultrasound. We see a mass here in the ear. And when we excised the tumor, it looked pretty big. Again, because of being a large animal model, ideally, when we transplant these cells, cancer cells into pig, the idea is that the tumor formed in these pigs will be bigger than the mouse model so that you have better utility. We ran some histology on these tumors, and we did a side-by-side experiment using NSG mouse. And all we can say at this point is that the formation of tumor is looking pretty different, as you can see, from the top versus bottom. They're different, and we don't really know why, but that's something we want to learn so that we can choose the best model to study particular cancer. So with all these studies, what we really concluded is that these pig models are not just a bigger mouse. It's not just, oh, there's a bigger animal that we use for for a purpose. But compared to a mouse model, they have very unique immune system. As you can see in this study, we compared a phenotype of RAG2 knockout mouse versus RAG2 knockout pig, and they're very different. So if you recall, I mentioned that we were able to induce teratoma formation in RAG2 pig model. That was not possible in RAG2 knockout mice. So the phenotype was different, and we think it's because of the presence of different lymphocytes at different maturation level. Again, that really implies that if you want to work with a model, you need to know the model, and that side is really underdeveloped. Well, pretty much beyond rodents. So pig, cow, and all these livestock models or farm animal models lack that information. So I would like to change the direction a little bit and see how having this technology really change or offer us a new direction. So now I would like to talk about how we can use the technology to elucidate gene function during early development. So network of genes are important for normal development. So as you can see from here, after fertilization and making a pig, that's a big step. So if we can understand the process, we may be able to generate better pig models or more pig models for food consumption. But all these elucidating gene functions are, most reports are based on rodent models, the mouse. There's no comparative studies to understand human development because, again, mouse just have a different developmental trajectory compared to large animal models. So these are just a few examples. So pig and cow embryos share key developmental events with humans, whereas mouse models don't. So these large animal models share demethylation patterns after fertilization. So after fertilization, dynamic changes in DNA methylation occurs to set the embryos for successful development. And timing of zygotic genome activation, which means the embryos are ready to use its own genome to make RNA. That's different in mouse and human, but these large animal models are pretty similar to humans. Expression of pluripotency gene markers are different, and amino acid metabolism is much similar in pigs compared to mouse. So what we wanted to do is there a way to disrupt a gene and look at the phenotype? So traditionally, conventionally, people used RNA interference technology. You may be more familiar with this technology as siRNA. It's pretty effective in silencing genes that are present in early stage of embryos. But now we started to utilize CRISPR-Cas9 system because we can modify a gene and the effect is permanent. What it means is if you look at here, so you inject siRNA into embryo when it's at one cell stage. As the cells divide, the effect of siRNA drops. Okay, so later stage, you may not see the effect of siRNA as good as the beginning stage. However, if we knock it out using CRISPR-Cas9 system, the impact should be permanent. So you can actually study the phenotype of embryos or fetuses, if you will, if you're interested in developmental biology. So this is, again, image of poking embryos. So we try to use this route to answer some of the questions during pig development. So one study that I would like to introduce is targeted disruption of TET1 gene. This was funded by USDA NIFA in US. So one thing we wanted to try was that, can we generate knockout embryos without having to breed these animals? Because that means that we need to generate animals. That's the first case, but then raise them to the age that they can breed, and then harvest embryos from a female. So each time that precious knockout animal might be sacrificed. So this is a really steep battle to do. So if we could just knock out these genes in embryos without having to maintain animals, that will be the ideal situation. So what we try to do is was to design multiple CRISPR-Cas9 system, and in part that was because the sequence information of pig genes are not really well known. So we designed multiple CRISPR-Cas9 system. We did an injection, and surprisingly, with the right design, we were able to knock out or disrupt TET1 in all embryos. So the knockout was very successful. All genotyped embryos carried mutations at the 3 target sites, and the targeting efficiency was 100%. So we moved forward with phenotyping, or looking at the impact of removing TET1 in embryos. And you can see here, when we disrupt the gene, you can see the development is a little bit lower. But the injection itself also had some compromising effect. So you had decreased blast formation, no significant difference in total cell number between TET1 knockout embryos and control embryos, which means that the formation, so the embryo may die from the injection, but the ones that are living have pretty much same cell number. So they're growing, well, at least based on cell type, cell number, it appears that they're normal. But when we look at their epigenetic status, the things were quite different. So when we looked at global level of 5-hydroxymethylcytosine and 5-methylcytosine in these TET1 knockout embryos, we saw decreased TET activity in TET1 knockout blastocysts. So when we looked at the overall TET activity, the activity was reduced, indicating that we have successfully disrupted the gene activity. And this gene is known to increase the level of 5-hydroxymethylcytosine, and we saw a decrease in 5-hydroxymethylcytosine. So that kind of matches our hypothesis, but we did not see any difference in terms of the level of 5-methylcytosine between TET1 knockout and control blastocyst. So again, these 5-hydroxymethylcytosine and 5-methylcytosine, they're a type of DNA methylation markers. So we can tell that by removing TET1, the level of DNA methylation has been altered. So next, we looked at the level of 5-hydroxy methylation and 5-methylcytosine by performing bisulfite sequencing, and we tried to capture these at a global level by amplifying or looking at DNA methylation level at repetitive elements. So we saw increased methylation level in PRE1 region, excuse me, in TET1 knockout blastocysts. So that's a euchromatin, and hypomethylation of central metric satellites in both knockout and control blastocysts. So because we didn't really see much methylation on heterochromatin regions, we weren't able to really see the difference, but you can see the euchromatin, actually the level of methylation was increased. And when we look at the transcript level of pluripotent genes in these blastocysts, you can see these, some of pluripotent genes were actually upregulated, meaning more RNA was present in TET1 knockout embryos. But these pluripotent genes were altered, but extra embryonic lineage genes, which means the genes contributes to placenta, they were not changed. And when we looked at gene levels or expression level of TET1, TET2, and TET3, we actually saw increase in TET3, which was a surprise, but our hypothesis is that the lack of TET1 activated TET3 to compensate its action. So therefore, we saw increase in the level of pluripotent genes. Later in this paper, although we did not use genome editing system, when we blocked the level or the activity of entire TET family, we did see a lower level of pluripotent genes. So again, by knocking out TET1, we saw elevated expression of NANOG and ESSRB gene, which they are pluripotent related genes, and then we saw threefold increase of TET3 transcript level, which presumably we assume that this was a compensatory action. So next, I would like to probably spend the rest of time talking about bovine studies. So again, as I mentioned, we mostly work with pigs, but we do work with bovine model. And when we try to use CRISPR-Cas9 system on bovine, interesting things happened. So application of genome editing tools beyond pigs. So CRISPR-Cas9 system is very effective in pigs. Often cases, we can reach 100% targeting efficiency, which means what it means is all embryos that we make, those are knockout embryos. How about in cows? They're farm animals, both large animal species. Can we use same strategy to disrupt genes in cow? So our biological question was, so this was a collaboration with Dr. Pete Hansen at University of Florida. So what he found was that by introducing a cytokine called CSF2, embryos were happier, he get a higher pregnancy rate. So our question was that then the benefit of CSF2, was it through its receptor or some other sources? Other actions. That's why we wanted to, or we designed to knock out its receptor, so CSF receptor alpha, and then try to see what happens. So we first disrupted CSF2 receptor alpha in pigs and cows. So pig knockout, so we used pig sequence first because we did not have access to bovine embryos at the time. So pig knockout, we knock out 5 embryos and they were all modified. It seems like, well, typical pig, we can disrupt the gene. So this is an image of successful pig modification. So it should be easy to apply to the bovine system. Well, that's what we thought. However, when we try to disrupt a CSF2 receptor alpha in cows, there was a high mortality. Embryo mortality after microinjection was 50%. So poking embryo was not as easy in bovine. So you can see here in this graph. So when we injected, so control embryos, so one did not get anything, was 20% embryos developed, but it's 70%, 7%, 8%, again, overall 26% versus 7%. So doing microinjection really harms the embryos. So that was the data. Then what, right? So we were like, okay, so microinjection is more difficult. What should we do? And also the mutation rate was well, much lower in cows. So we actually used identical CRISPR sequence between cow and pig, but the efficiency was, well, not even 50% in cow. So that was a very disappointing result, but really made us appreciate the value of each animal, well, each species. And we really need to put an effort to optimize and design a system that are customed for each species. So we decided that, okay, species-specific optimizations are needed. So we really started over. So we customized this for bovine. So we redesigned all the guides and we tested everything on bovine samples. We just excluded the fact that, well, our assumption that if it works in pig, it's going to work in bovine because, well, we learned it doesn't. So we designed a small guide, single guide RNAs to induce intended deletion of exon 5. And another reason why we did that was that there was a presence of a pseudogene on bovine genome that are pretty much identical to our target gene, CSF2 receptor alpha. So the only way for us to design a CRISPR system that won't disrupt the pseudogene was to design this CRISPR system expanding introns and exons. And then we had to optimize the concentration. So we did a lot of experiments, tried to optimize the system. So here's the result. So when we actually use an optimized system, so that are the system that are optimized in cow, And then we really wanted to see what happened. So we actually simultaneously genotyped genomic DNA and the messenger RNA in these embryos. And we were able to reach 100% targeting efficiency. Again, I mean, this table really summarized, well, it doesn't really reflect the amount of effort that we had to put into to reach there, but after all this genotyping, we felt comfortable that we can disrupt this target gene in bovine. So this is the result of our bovine experiment. So we targeted CSF2 alpha receptor alpha in cows, and you can see here, let's see, so there was no, again, drop in development. So we also changed microinjection approach. So we added some chemicals that will help us reduce the mortality, and you can see here, so compared to the control and even IVF control where it did not receive anything, no microinjection, their developmental trajectory is pretty comparable. So again, that really make us wonder this each species is unique. And those embryos were used to analyze the signaling pathway, which I don't have time to go over it at this point, but you can see here this paper we published in 2021. So with all these examples, so what can we do now using genome editing? So we can disrupt multiple genes. So multiple genes can be simultaneously modified during embryogenesis. Immunodeficient pigs can be used to generate patient-derived xenograft models. So these will be great resource for regenerative medicine. If you want to introduce stem cells or mini organs, that should be a great application. And we can use these pigs to grow human tumor for cancer research. Knockout embryos can be effectively produced without having to establish knockout livestock animals. Again, generating these animals and breeding requires a lot of time and effort, but by introducing CRISPR-Cas9 system directly into embryos, we can bypass that issue or problem. Study events of early embryogenesis using livestock species is also possible, so we can get more quality information, just to maybe basic to understand the phenotype of animals, but also it can reflect to human phenotype. Shortcomings that I did not talk about today is specificity, toxicity, and optimization. Optimization, I said a little bit in the cow study, but this is not really a simple study where you can pick a condition and just use it, but the technology is not there yet. This is just ongoing and future directions in my lab. I still, again, the core of my program is biotechnology, embryology, and reprogramming. For example, under biotechnology, we try to continue to develop a safe approach to use genome editing system and better pig models for cell transplantation. So we are currently designing and producing different immunodeficient pigs to enhance the outcomes, improve efficiency to produce pig models in general. And this is related to the TET1 knockout embryo topic, but we are interested in understanding mechanism of oocyte-mediated reprogramming because, again, there's a dynamic change in DNA methylation after fertilization, and that process is necessary for normal development, and that's where we want to understand the process. And strategies to produce healthier in vitro-derived embryos. In vitro produced embryos are typically considered suboptimal or sub-quality compared to in vivo counterparts. But hopefully by improving culture techniques and in vitro maturation techniques, we can improve their quality. Last slide, this is acknowledgements. My current lab members, 2 postdocs currently working on the various topics, and students, technicians. And working with pig in general, it's a big task because, well, simply pigs are big, so that's been a struggle. But I have different collaborators helping out with the topics, NSRCs, Swine Center. All these people help me out, and I have different collaborators helping me with immune profiling or surgical technology or providing organoids. Lastly, and this is well, before I go last, this is an image of my current lab, and we're actively recruiting all the time. So if you see yourself working with pigs or pig samples or trying to develop a unique genome editing technology, please reach out to us. And lastly, again, all these research activities won't happen unless we have funding to do so. And fortunately, the program has been funded by USDA and NIH and other collaborators, University of Florida or Cincinnati Children's provided funding to get this study done. With that, I will stop my presentation or seminar here, and thank you for the attention. And if you have other questions, please reach out to me. You can find my information at try to. Oh, sorry. Try to turn this off. Not working. But anyway, so thank you for your attention, and I will stop here. Thank you.